Hello everyone, this is Latha Akula. In this video, we are going to study about different phases in the mitosis. Actually, we have completed in the last video interphase, right? In the cell cycle, after interphase, what is the next coming phase? Mitosis. In the M phase. In this M phase, we have studied about mitosis and meiosis. In the mitosis, there are four stages are present: prophase, metaphase, anaphase, and telophase. In that, the first phase is the prophase. So, what is meant by prophase? Pro means first and phase means stage. Because this prophase is the first phase, we are calling it as prophase. And here, interphase is followed by mitosis or M phase. In that, the first phase is the prophase. And here there are some changes occurs in the prophase. What are the changes? Condensation of the chromatin material occur. What is meant by chromatin material? In the cell, within the nucleus, the genetic material was in the form of a coiled thread-like structure. That coiled thread-like structure that starts coiling and form a chromosome structure, right? So here that condensation process occurs, and after condensation what it will form? Chromosome. It forms the compact mitotic chromosomes are formed here, and these mitotic chromosomes, how they appear like? They just appear like a ball of woolen ball like structures, and that stage is called as spireme stage, because the chromosomes will not properly arranged. It was just like the ball of wool, and that stage is called as a spireme stage. And if you see in the animal cell, there are a pair of centrioles present above the nucleus. The duplication of those centrioles already occurs in the interphase. And here, those duplicated centrioles, they start moving to the opposite poles. Here, there is a one pair of centrioles, and here there is another pair of centrioles will appear. From those centrioles, the spindle fibers arises. The initiation of those spindle fibers also occurs in this prophase. And then at the end of this prophase, the several cell organelles like the Golgi complex, endoplasmic reticulum, nuclear membrane, and nucleolus, they will not seen in the cell. This is about the several features shown in the prophase, and the next video we are going to study about metaphase. In this video, we are going to study about the second stage, that is the metaphase. Here, meta means second, phase means stage. After prophase, this is the second stage, hence we are calling it as the second stage or the metaphase. And here what happens? What are the features observed here? The disintegration of the nuclear envelope occurs. Already in the prophase, we have studied that disintegration of the nuclear envelope starts. Here the complete disintegration of the nuclear envelope occurs, and the chromosomes, they spread in the cytoplasm. All the chromosomes, they have been spread in the cytoplasm, and if you see the chromosomes, they have two chromatids, because after replication how many chromatids will be seen? Two chromatids, right? Here it has two chromatids. They have been joined by means of a centromere. By means of the centromeres, two sister chromatids they have joined. Next one is the chromosomes at this metaphasic state. They were thickest and shortest. Hence, in this stage, the morphology of the chromosomes that can be easily studied. And here, the mitotic spindle formation completes. Here, the mitotic spindle formation is what? From the centrioles, the spindle fibers arises. The initiation of that spindle fiber formation occurs in the prophase. Here, in this metaphase, the complete process occurs. Here, the metaphasic spindle formation is completed in this phase. Next one is chromosomes. These chromosomes are spread here, right? All these chromosomes, they reach to the equator of the spindle. So all these chromosomes, they reach to the equator of the chromosome. The phenomena of reaching all the chromosomes to the equator of the spindle is called as congregation. And after reaching to the equator of the spindles, the chromosomes, they align themselves at the plane of spindle fibers at the center and form the metaphasic plate. Here the metaphasic plate will be formed. And later what will happen? How these chromosomes attach to the spindle fibers? by means of a special disc-shaped structure called as kinetochore. If you see the chromosome, there is a central, there is a centromere. By means of that centromere, the two chromatids have been joined. And at the surface of the chromosome, there is a disc-shaped structure is present, which is called as kinetochore. By means of that kinetochore, these spindle fibers arising from the centrioles, they join with the chromosomes. How the chromosomes attach to the spindle fibers? By means of kinetochore. Kinetochore only. And what are these spindle fibers? These spindle fibers are nothing but microtubules. So these microtubules will study in the cytoskeleton. Already we have completed this cytoskeleton topic. In the cytoskeleton, we have studied about microtubules. So these spindle fibers are nothing but the microtubules. These spindle fibers, they were attached to the chromosomes by means of kinetochore. These are the features seen in the metaphase. What are the features we have studied? Complete disintegration of the nuclear membrane occurs. The chromosomes spread in the cytoplasm and the mitotic spindle fibers formation occurs, and the chromosomes, they align themselves at the center, or they align themselves at the equator of the spindle fiber and form the metaphasic plate. These are the important features we'll study in the metaphase. And the next one is the anaphase. Phase. Here ana means up, phase means stage. What happens in the previous stage, what we have studied, all the chromosomes, they have aligned themselves at the center of the cell. They form the equatorial plate. But in this phase, the chromatids, they move towards the poles in the upward direction. The chromatids, they move to the opposite poles of the cell. What happens? The centromere splits. So here, if you see this is the chromosome, right? At the center, there is a centromere. By means of this centromere, only two daughters have been attached. In this stage, the centromere will split, and this is one chromatid, this is another daughter chromatid. These two daughter chromatids, they have been pulled to the opposite direction. See this? Here the chromosomes, they attach to the spindle fiber by means of the kinetochore. At the center, there is a centromere. This centromere split in this, and the spindle fiber, the length of the spindle fibers shortens. Because of the shortening of the spindle fibers, the chromatids, the daughter chromatids, they have been pulled in the opposite direction. All these daughter chromatids, they have been pulled to the opposite direction against the equatorial plate, and they reach to the opposite poles. During that moment, there we see the interzonal fibers. So these are the interzonal fibers. So in which stage we will see the interzonal fibers means what we have to say? Anaphase. And here, during the movement of these chromosomes, or during the pulling of these chromosomes, we will see the different shapes of chromosomes. And this is the best stage to study the different shapes of the chromosomes, like V shape, L shape, J shape and I shape. There may be in the Neet, there may be a question like, what is the best stage to study the different shapes of the chromosomes means what we have to say? It is in the anaphase. So, in the anaphase, what happens? The chromosomes at the center, the centromere splits, and the two daughter chromatids will be separated, and there may be the length of the spindle fibers decreases. Because of the decrease in the length of the chromosomes, the chromatids will be pulled to the opposite poles. The centromere starts moving to the opposite poles. Along with that, the chromatids also moves. So this is about the anaphase, and the next one is the telophase. First phase in the karyokinesis is telophase. The mitosis, there are two phases: karyokinesis, cytokinesis. Karyokinesis means that the nuclear material will divide it. Cytokinesis means the cytoplasmic material divides. In the karyokinesis, prophase, metaphase, anaphase and telophase are the four stages, right? What is the last phase of the karyokinesis? Telophase. So, in the telo means end, phase means stage. This is the last stage in the karyokinesis. What happens here? Already in the anaphase, what we have studied? The chromosomes, not the chromosomes, the chromatids, they pulled apart to the opposite poles. The spindle fibers shortens, right? The chromosomal material that will be pulled to the opposite poles. Here in the telophase, the chromosomes, they reach to their respective poles. After reaching, what happens? The mitotic spindle disappear. Whatever the mitotic spindle fibers that were attached to the chromosomal kinetochores, that disappears, and at the same time chromosome, they start uncoiling. What is meant by uncoiling? The chromatin material that will coil and form the chromosome. Here again the uncoiling process starts. The chromosomes that will become into the chromatin, and here the uncoiling of the chromosomes that leads to the thin, slender, long mass of chromatin formation occurs. Here the decondensation of the chromosomes occurs. Here the chromatin material, when it is condensed, it forms the chromosome. Again these chromosomes, they will again decondense and form the chromatin. Here the mass of chromatin is formed because of the decondensation of the chromosomes. That means the chromosome, they lost their identity, they lost their shape, and again that will become into the coiled thread-like structure. And here whatever the cell organelles disappeared in the previous stages, that will again reappear, and that is nucleolus, endoplasmic reticulum, Golgi apparatus and other organelles that will be reappear in the daughter cells. And the nuclear membrane which have been disintegrated in the prophase, that will reappear in this telophase around the chromatin. Around the chromatin, what is formed? The nuclear membrane formation takes place. And next to this telophase is, it is followed by cytokinesis.